times.
Okay.
And I believe you're speaking for 50 minutes, is that correct? Or are we going to get a duet halfway through?
Did you say 50?
It's down to 15, you're down to speak for 15:40, you see, so and Alan's going to start halfway through. So.
All right. Okay, well, I'll be talking about the isomerization of fluorescent protein chromophores. Some of this work was done in Queensland. Some of the work I'll be talking about was done during my PhD thesis in Illinois with Todd Martinez. First off, an introduction to the fluorescent proteins. I don't know how many cell biologists there are in the audience, but fluorescent proteins are very well known to cell biologists because they're very useful. Their biological background as a family of proteins is as follows. The first one to be discovered was the green fluorescent protein, which is reasonably well known. So they're sometimes called the GFP homologs, and some people call them all protein chromophores. I call them fluorescent proteins, although not all of them are fluorescent. They're a metazoan superfamily, which is now known to span quite a large, to span several branches of the tree of life. Particularly well known in cnidarians, which is to say jellyfish, sea anemones, and coral, but also more recently discovered in bilaterian organisms, such as, for example, this copepod here, which is an arthropod, and there have been some homologs identified even in our own phylum. All of this has happened since about 2000, I think. So in the last 7 years, we've gone from Hydromedusa, which is there, but to a large amount of cnidaria and also some other species. The proteins are of interest because they're very useful. As I'll mention soon, they become fluorescent. When you express them, so all you need is the gene in order to have a working fluorescent probe. They're useful in microscopy and all sorts of imaging applications, including including constructs that you can build that use forced or resonance energy transfer. You can use them for in vivo imaging, particularly those which occur in the red region where the absorption cross-section of biological tissue is quite low. And they can be expressed in a range of organisms, including mammals, but also including insects, amphibians here, all sorts of single-celled animals. They're extremely useful. And those of you who follow Ainley might know that actually the Hulk is green because he expresses GFP. There are now, of course, since 2000, a wide range of different rainbow Hulks that Stanley could think of. The structure of all the proteins is what's known as a beta can because it looks like a can of soda, and the sides are wound around with beta sheets. They're capped with helices. There's a helix that goes down the center, which you can't really see in this figure. And this helix is broken. In the middle of this break is the chromophore, which is actually responsible for the light absorption and emission, gives the protein its optical properties. This is buried deep on the inside of the protein, where it's surrounded by a somewhat unusual number of ionizable residues for the interior of the protein. The entire protein itself is usually around 230 amino acids, depending upon the variant. And the chromophore forms automatically after the protein folds, which is why the protein is useful and why you only need the gene. So if you express it, you get a fluorescent marker. You don't need to add any exogenous chemicals or dyes. The chromophore forms from a tripeptide sequence, which is always something glycine, where the something in the middle is usually tyrosine in most naturally occurring forms, although mutants have been engineered with tryptophan, phenylalanine, and more recently a wide variety of unnatural amino acids, which produce new proteins with optical properties. But in most functional natural varieties, it's tyrosine. The first something can change quite a bit, and sometimes you get different chromophore structures depending on this. But the basic chromophore structure, which forms, and which is a common motif in all the proteins, with the tyrosine in the middle of the tripeptide, is a parahydroxy benzylidene imidazolinone moiety. It cyclizes, the protein chain bites itself. Then there's a 1,2 oxidation, which forms this conjugation in the bridge, and you end up with a conjugated pi system, which absorbs in, depending upon the particular varieties of chromophore somewhere in the visible and a bit somewhere to the red or the Azores. Once you form the parahydroxybenzylidene midazolinone, you can have downstream reactions which actually produce slightly different chromophores. And this slide is a bit old. At that time, I was pointing out 3. There are now known to be 6 distinct chemical structures which can form a chromophore in GFP, or pardon me, in FPs, for now all of them are green. GFPs actually have just the double-membered, double ring structure. They can become anions, and most functional GFPs actually, or FPs, again, are anions. If this protein backbone linkage, because the whole thing is connected to the backbone here and here, in GFP, if this then becomes a double bond, then you get an N-acylamine. These proteins are red. Those that are fluorescent, those that are not fluorescent, they're usually bright blue and purple. This structure actually can then hydrolyze in certain conditions in certain mutants to produce a chromophore that looks like this. These chromophores are most known for a reversible photoactivation of fluorescence, and they're sometimes called kindling fluorescent proteins because of this. Another interesting variant is KDA, or are the red to green photoconverters, which are exemplified by KDA, or EOS-FP. These proteins have a histidine in the first position of the tripeptide, and when you shine, they're green until you shine intense blue-green light on them, at which point they photoconvert and undergo additional reaction. They form double bonds here, and then they switch to red. There are orange, this family of proteins which are orange, which were recently identified in another chromophore, a yellow one. There are 6 in total. At the beginning of the conference, the concept of Rosetta Stone was mentioned, and that perhaps one of the goals of this workshop is to identify Rosetta Stones. I would put to you that FPs are a very useful Rosetta Stone because they have a very wide variety of photochemistries and a very wide variety of features for a family of proteins with a homologous structure across the protein family and a reasonably small range, again, 6 chromophores, all of which contain a common central motif. So the emissions can go from about 450 for a cyan fluorescent protein down to 650 or up to 650 in wavelength for the most red fluorescent proteins that have been discovered. The quantum yields can go up to 0.8, but they can range all the way down to undetectable. And in some cases, you can have, for the cost of 1 or 2 mutations, something that goes from 0.7 or 0.8 all the way down to undetectable. They're easy to produce because all you need is the gene. They're usually, well, all the ones that I know of are soluble, and they're usually stable with respect to temperature and denaturients. And within the superfamily, you see a very wide variety of photochemistries, including the quantum yield, including the range of emission, but also reversible and irreversible fluorescence activation, singlet oxygen generation. In fact, there's recently been a protein that was engineered to create singlet oxygen with such ease that you can express it in cells and then burn them away with a laser. This is called killer red. Excited state proton transfer occurs in several of these proteins. Photoactive color changes, as I mentioned earlier. And they often like to form oligomers where you have different barrels sticking together. If you have then barrels with appropriate emission and absorption, you can get fret transfer between monomers. You can use this to make constructs where fret turns on and off in response to binding. Things like this. So there's a lot of things that you can investigate in the context of these proteins that are very interesting in this way. So what I'm now going to talk about, as the title indicated, is one particular facet of this very multifaceted story. I'm going to talk about internal conversion. And accumulating evidence seems to suggest this is a major route of radiationless deactivation of internal conversion in the protein family. It's interesting to understand because if you want to engineer, hopefully rationally engineer new proteins, hopefully do it on a computer, because that's what I do, then you want to understand the process by which they come to fluoresce or not fluoresce. And so the data that suggests that Cis-Trans isomerization is a major internal conversion route in these molecules includes the fact that most FPs, if they are denatured, do not fluoresce, and model chromophores usually do not fluoresce. There's, I think, one notable exception that I know of, but they usually do not. The data for synthetic models is consistent with fast isomerization via a conical intersection, and the time scales are reasonable for Cis-Trans isomerization of the exocyclic bridge here. You can generate fluorescence FPs or from non-fluorescent FPs and vice versa, and the mutations that you have to introduce. Some of the major mutations are in places where you would expect, or at sites where you would expect stabilization of a particular cis-trans form to where you would expect a particular form to be stabilized. Non-fluorescent colored proteins, chromoproteins that absorb, do not emit, tend in their crystal structures to have non-planar chromophores, which supports an isomerization process because in a two-mode model of isomerization, you would not get fast internal conversion, or you'd get fast internal conversion via this route if you actually had a non-planar chromophore to begin with. And theory that I performed and others supports the contention that the S1 state lowers its energy when you twist, and that there are low-lying conical intersections which correspond to twisted chromophores. So this is probably a review for everybody, but when I talk about extreme on the potential surface of these chromophore models that I've calculated, I'm going to be talking about multi-state mechanisms to extend what one learns in first-year organic chemistry to excited states. The basic thing one has to do is to realize that for each electronic energy you have a different surface. As on the ground surface you have reactant basins, product basins, transition states. You also have these on the excited state, but you also have a new feature which is known as the conical intersection, which has been mentioned a few times I think in this conference. You can roughly think of these things as transition states between different electronic states, but there are important differences, one of which is that they're not necessarily well-defined points, they're extended seams, because here the coupling and the degeneracy of the states, the diagonal degeneracy and the coupling, both have to go to 0. These are 2 parameters. So these can actually be seams of the full dimensionality of the system minus 2. When I talk about a conical intersection structure, I will be talking about a minimum energy point on the seam. So, to briefly go into what I'm going to say about photoisomerization, let us consider for a moment ethylene, which is the hydrogen atom of photoisomerization reactions. Now, it's widely, evidence is growing that in order to really understand these reactions, you need to consider 2 adiabatic states, but 3 diabatic states, which correspond to the ground state singlet, the excited states, covalent charge transfer states, and the ionic states too. In order to reach the conical intersection, which mediates isomerization in systems like this, you eventually have to break the symmetry. So ground state ethylene has D2H symmetry, and if you twist it, but then you also have to bend one of the carbons. This breaks the symmetry, which tunes the site energies, which corresponds to the diagonal degeneracy tuning that I mentioned when I was talking about conical intersections. One important thing that comes out of these analyses is that the intersections occur in charged localized states. So, for example, here are shots from a trajectory of ethylene performed by Jay Quenville several years ago in Martinez lab. He's now at Los Alamos. 0.2 you have planar ethylene, which is promoted in the excited state. You progress, you bend, you twist. You end up with localized orbitals corresponding to different hybridization state of the carbon, which can hold an anion charge. And if you calculate the potential surfaces of neutral or cationic GFP chromophores, you find that the situation is similar. The bond that is formally a double bond in the ground state wishes to isomerize on the excited state. The bond that is not formally a double bond doesn't wish to isomerize on the excited state, so that's not favorable. Over here somewhere you find conical intersections which are twisted about the relevant bond, which may show parallelization of the hydrogen, but in all cases along at about 45 degrees of twist, you start to have charge localization across the twisted bond with the sense of the charge localization switching from the ground to the excited state. To extend this to an anionic GFP chromophore, one needs to realize that for an anion, then you deprotonate at this site, you have a negative charge here, you can also have a negative charge here. There's nothing that really prevents us from drawing this other resonance structure, so you have a charge transfer resonance between the forms. To go to the hydrogen atom of coupled two-bond double bond isomerization, which would be an allele anion, you would then have configurations which look like this. A similar story comes out of it. From this resonance for the GFP chromophore, because this resonance is so strong, you end up in the excited state, inverting the resonance, or alternatively having a superposition of different diradical forms. Each of these can isomerize. As the isomerized charge separates, the sense of charge separation depends upon the ground in the excited state, so it's reversed going from the ground to the excited state, and it also depends upon the bond along which you isomerize. So if you isomerize about this bond, then you end up separating charge such that on the excited states, you localize electron density on this side, or on the ground state you would localize it here. Actually, I guess I wanted to do that up here. If you isomerize about the phenoxy bridge bond, you end up on the excited state localizing electron density here in line with the resonance structures that we've drawn. A opposite situation occurs if you isomerize about the other bond. I mean, there's only no bridge bond, in which case you end up. Right, yeah, on the ground state you localize it here for this one and off here. Sorry, I was going to keep you straight. But the point basically is that if different bonds isomerize, then the charge localization is opposite. So the pathway determines the charge localization. Intersections occur where these charge localized states meet. Here's one of them. This was calculated with Cas 1211 wave function for a red fluorescent protein chromophore model, which actually happens to have an intersection at the bottom of the excited state surface. So it looks like this with the branching plane, which is this. These are the 2 degrees of freedom, which you lose out, everything else being degenerate. So they intersect, and actually at the intersection, the sense of charge localization switches if you go across the intersection in one of the directions which define this plane, which corresponds to a bond length alternation. And this is the tuning coordinate. So there are charge-separated states in the photoisomerization of fluorescent protein chromophores. And the conical intersections arise when states of different charge separation sense meet. You can tune then the surfaces. You can extend this reasoning to understand how substituent effects affect the surfaces. For example, I've already told you that there are 2 pathways for GFP chromophore, but for an RFP chromophore where you have an acylamine, which is strongly electron withdrawing, you can tip the scales and selectively stabilize one path over another. And a good example of this is to take a model RFP chromophore with an N-acylamine, which is electron withdrawing, and switch the acylamine for an olefin, twist it, and see what the potential surfaces do. For this case, we're twisting here about the phenyl bond so that the electron density in the excited state wants to go over to this side near the acylamine. You raise the ground state, you lower the excited state, you end up running into a conical intersection seam at about 65 degrees. And actually the bottom of the excited state is the conical intersection. Thank you. For the olefin case, this doesn't happen. You still have a separation at twisted geometry. Okay. So you can do dynamics on it. Here's a dynamics running in vacuum. We can talk much about it. But you can do this. We've done it with a empirical method, parameterized against the ab initio data. You can then put that in solution, and what you find is actually that in solution, the isomerization rate goes up drastically, so the population dumps the ground state faster. This is actually consistent with experimental observations in solvents of different polarity. So here's the vacuum data. Here's the data in a small water cluster in vacuo with about 53 waters, I think, around the GFP chromophore. The waters act to stabilize the charge transfer states, and this accelerates isomerization, which is shown here. So if you have, for example, if you were to have a covalent ground state and a charge transfer excited state, if you put it in a solvent, you lower the excited state, you move the position of the intersection, you can even make it the absolute bottom of the excited state surface and very much speed up isomerization. And you can do it with static charges. So, for example, going back to ethylene again, the hydrogen atom of double bond isomerization. This again was done by Jason Quenville at the Martinez lab several years ago. If you plot the surface, bending one carbon and bending the other carbon, then you put a charge near one end, you can actually tune positions of the intersection. And so then the question which arises then is, okay, I've told you a lot about chromophores, but what about the protein? And I'm sorry about that. And there's been a lot of talk about what exactly it is in the protein that stops isomerization in proteins with fluorescent. And there have been some models proposed. Mark Zimmer has done a lot on this with molecular dynamics models, and it's been put forward that perhaps there's a hula twist mechanism involved and it sweeps out this volume. I don't know really if I believe this story because, as Dr. Hayashi was pointing out, photoisomerization is multidimensional. So you don't really have this thing twisting around and sweeping out all this volume anyway. And you can find hula twist motion as a kinematic effect in vacuum simulation. So I don't know if I buy this. The results of this study, what they showed was that if you twist about the phenoxy bridge bond, or if you do a hula twist, you can make it to the intersection, or you can twist the molecule, but around the imidazole bone bond you can't. This is interesting because it raises the prospect that the path influences the decay rate, but it's contradictory with certain observations, such as the fact that neutral GFP chromophores and neutral FP chromophores in general have lower quantum yields. They all would want to isomerize about the bond, which these simulations say you can't isomerize about. Whereas anions could go both ways, and RFP chromophores, for example, the substituent effects favor the phenoxy twist, so they should actually not be fluorescent, but some of them really are. And in fact, you can try, well, we did at one point try in Illinois, we attempted to make a steepest descent pathway for GFP, actually using, again, this semi-empirical model in a QMMM formulation. This was very late in my PhD. And we found actually that there was a barrier to isomerization, whereas in vacuum there was not, in water there was not, and we actually stabilized the intersection. Unfortunately, I still can't tell you what the physical origin of this barrier is. But what I would like to suggest to you is that it may not be steric control, but electrostatic control is also a possibility because of the charge transfer intersections that you see in the system. Okay, and that's about it. We're hoping actually to, I'm amassing a big data set. We hope to parameterize a simple model and get some good statistics. I'd like to thank everybody on this slide, particularly these people who are not here to properly be congratulated with some of the data that I
Okay, thanks very much. Right, are there any questions for us?
Are there any indications that there are specific hydrogen bonding interactions in the chromophore in the protein?
Yeah. Yes, actually.
And are they simulated in these calculations? In other words, hydrogen bonding interactions can change the charge distribution.
Yes, they were. But the hydrogen bonding interactions crossed the QMMM boundary, so there's a whole host of questions that come about because of this. If one assumes that hydrogen bonding is always primarily electrostatic, then perhaps it's a fair assumption. Although then you're also treating things as point charges, and the QMMM is sometimes, there are a whole lot of questions buried beneath the surface about the boundary. So yes, they were, but no, I can't tell you exactly what that means right now. You would have to compare that against a larger calculation, and I didn't have time to do that before I got out of Illinois. But yeah, the mutations that switch fluorescence actually are often at hydrogen bonding sites which would tether the chromophore.
I said one question. This is very interesting because originally always opposite to people thinking because in actual the static issue, the electrostatic and the static issue. So what do you think that this in the solvent you lost the fluorescence? So if we electrostatic, you really means it's a heterogeneity static problem. It's not the same local polarization issue. Because I think if you put the water, put the very high non-cold or whatever, if you can dissolve, you be electrostatic issue might be significantly different for this chromophore. So is there any data, which I forgot in my mind, is there any data show this chromophore in the, let's say, water and in, change the dielectric constants, say the quantum yield is a significant difference or very similar?
It is different. Yeah, you can, if you put chromophores in nonpolar solvents, then the quantum yield does raise.
I see.
Perhaps not that much, not as high as does the protein, but on the order of maybe 0.1 to 0.4, I think, perhaps. Whereas in water, for mild GFP chromophore, it's undetectable.
So in terms of, you said that sort of the anions with the fluorescence, they have to be the anion to be fluorescent.
They tend to be more fluorescent.
So, and in your isomerizations, you show various sort of intermediate structures, which can be diagonals or whatever. But actually, one of them could be just a straight phenol structure. So, do people have any idea of the structural chromophores, if you like? Do they have carbonyls, which is a quinoid on the phenolic end, if you like, and then effectively the amide end has a stretch of the carbonyl, which is sort of a single bond character.
Do you mean that these been synthesized?
So no, just sort of measured. Go back.
This is what you mean?
Yeah. So that sort of structure there. Is there sort of evidence, physical evidence from infrared spectroscopy, whether you're sort of sitting in this resonance form or you're in that resonance form? And does that give you ideas about why something is fluorescent or is not fluorescent?
I don't think so. There have been transient IR. There was a transient IR study of a model chromophore done. In fact, if you put this whole thing in solvent, then it tends to stabilize this form actually. Or in PCM solvent. Studies which have put, or studies which have tried to model the protein, however, don't show this. And in fact, there's an arginine sitting here.